I want to thank Mike and the group for inviting me to talk. As Steve said, is that usually I get a call from the nutrition group and they say we need pestilence. and that's my job. This is not usually a group of people who I talk to, but I do recognize two people that I want to give a shout out to. One is Steve Henry, and the other one is Lisa Tokash. If anything, they have given my work a sense of purpose, if not a sense of urgency. If you're used to working with Steve Henry, he calls you up and says, so what have you cured for me today? And it's usually not just a rhetorical question, it's usually a request. So with that, and Jim Nelson, I talked to him earlier in the week and I said, Jim, yeah, absolutely, I'm going to do two things. I'm going to show one slide and I'm going to talk for less than 30 seconds. Okay. So here's the one slide, Jim. I've worked on this disease for more than 20 years, and as the slide says, we finally have a cure for Prrs. So for the next 14 minutes and 30 seconds, I'm just going to... No, I probably should tell everybody how we got to this discovery. First of all, my work is not done in a vacuum. What I'm going to be talking about is some of the genetics work that's done in my lab, and it's done with a group of collaborators. I want to recognize Randy Prather at University of Missouri, who makes the genetically modified pigs that I'll be talking about. Jack Deckers at Iowa State, who has done some incredible genomics work related to Prrs resistance, and a long-term collaborator, Joan Lunny. Again, I do for these people what I do for the swine group. I am the pestilence. They make the genetically modified pigs, or they give me the gene, and I provide the virus to test it. So this is Prrs. It was really first described by Kerry Kefauver in 1989. And it was described as an influenza-like illness, mid to late term abortions, high pre-weaning mortality, and poor growth performance. That description of Prrs sticks. I think it's still how we see it today in 2016. In our genetics work, we recreate populations of Prrs infected. We create Prrs infected populations. And this is really how we see Prrs. If you just take pigs over a period of time after they've been infected with Prrs, and you kind of just weigh them. And you look at this weight distribution. And if you look at these black squares here, these are uninfected pigs. And you can really see what Prrs does at the production level, especially the isolates that we use. We use isolates that tend to be a little more pathogenic than what you might find in the field. And so what you see is that in our experiments by day 42, the population distribution just falls apart. And in fact, these pigs, which we call laggers, will eventually catch up, but it requires a little more time on feed, and that is probably one of the major costs to the industry. If you want to put a monetary cost on it, since its appearance, it's probably cost the industry about $14 billion. That's significant when we look at an industry that depends on small profit margins in order to be successful. I always say Prrs is not a pig disease, it's a production disease. And we see it as an endemic disease in a population of pigs or a herd with periodic outbreaks of severe disease. It has some important properties, and one of them is persistence. It's able to persist in a production system by being very efficiently transmitted, not only from pig to pig, but also from mother to fetus. We've done a lot of work, and you can see it's persistent in a population, and it can persist in a pig for more than 100 days. So in the case of Prrs, this is really the big bugaboo about this disease. The other thing I want to point out, it's stealthy, easily transmitted, and another way that we see it is that it participates in polymicrobial diseases. You can live with Prrs, but you may not be able to live with Prrs and myco. You can live with Prrs, you may be able to live with PRRS and myco, but you're not going to live with PRRS, myco, and influenza. So when we look at, well, what does PRRS cost? We always talk about animal health. I don't care about animal health. I care more about nutrition, because feed is the greatest cost that the producer has. So when you look at PRRS, you can really look at it in wasted feed. Feed that's wasted because you have sick or dead pigs, pigs that are slow growing, and also this energy cost for these secondary infections. And so we can kind of expand this picture, and we can talk about PRRS having a nutritional impact, but also an environmental impact. This wasted feed has to go somewhere. It's going to go into the environment. And then beyond that, we can extend this into societal impacts. About 10 years ago, I rethought PRRS. I don't care about the virus. I don't care about the virus. The only thing that I know is, what does this virus take from you, and how do you get it back? And so over the years, my lab has really taken a three-prong approach. And the idea being is, how do you get these pigs to move up into these higher weight classes? And so in my lab, we use a lot of the traditional disease control techniques, such as vaccines, better detection, biosecurity, and epidemiology. We've kind of moved into the nutritional area, and you're going to hear some work by Megan Niederwerder. We think in terms of the effect of PRRS on feed efficiency. Also, how do we develop better feed formulations to feed that PRRS infected population? And then Megan is going to talk about the microbiome. What I'm going to be talking about today is genetics. And again, the only thing that I'm concerned about is, let's get back this 10% that PRRS takes. I don't care how you do it, what type of combination of tools that you use. And that's really what kind of got us into pig genetics. What can pig genetics do for us? Not only looking for pigs that are resistant, but also looking for pigs that are tolerant. Another word that geneticists use is resilience, which I don't know what that means. And then whether or not we have pigs that are vaccine ready. Obviously, genetics controls a lot of things, and it's going to control your response to a vaccine. And then finally, I'll be ending my presentation just saying a few words about the newest technologies, which is genome editing. We've had PRRS vaccines since 1994. They haven't changed. There's the same modified live vaccines that we've had on the market for the past 20 some odd years. Vaccines do derive a benefit. However, they do come at a cost. And one thing I can say about vaccines, the conclusion is that vaccines are a poor option for disease control and eradication. Vaccine is not an eradication tool. And also, one thing that we learn is that even if you have a pig that's vaccinated, it cannot be transported to a PRRS-free region. In other words, exports to countries that are free of PRRS will not accept pork from vaccinated animals. So what we have been doing for the past 10 years or so, working with folks at Iowa State, is really to look at some of these genetic tools that we may be able to use to improve the cost of PRRS. And some of these tools are going to be marker selected breeding. We've done a lot of genotyping, something called genome-wide association studies. I won't explain what they are because I don't understand them. Okay, and then finally, actual gene modification. In other words, if we find a gene or a mutation that's beneficial, can we actually genetically engineer it back into the animal? And in this case, I'll be talking about the deletion of a gene that's involved in PRRS susceptibility. Some important findings from this genetics work is that approximately 40% of how a pig responds to PRRS is related to pig genetics. That's a big chunk. And so what this says is that we can now begin to think about breeding pigs that have improved disease resistance. And some of this work in collaboration again with Jack Deckers is that there is a favorable genotype in which if you're a pig and you have PRRS and you have this favorable genotype, you're going to get about 10% increase in weight. Okay, remember how much PRRS was taking from me? About how much? 10%. Now I can start to get it back. And if you have this favorable genotype, you're also going to have less virus. But I want to kind of change gears and talk a little virology, and that is to describe how a Prrs virus gets inside the cell. It's a very simple mechanism. It's a lock and key. You can imagine that this is the Prrs virus, and it has this key, and it's going to insert this key into the lock, and that will be the way in which it opens the door and gains entrance into the cell. This is how we learn virology, and guess what? This is how I teach virology, okay? And in this case, when we look at this keyhole mechanism, we're actually looking at a surface receptor on a cell we call CD163. So if you can think of just CD163 as being this keyhole, what happens if we get rid of it? If we prevent the virus from being able to insert its key into the lock, can we block infection? And so we do this through a mechanism that we call gene modification. The revolution in gene modification is something called CRISPR-Cas9. If any of you have listened to the radio or watched TV in the last 6 months, you will not know, but you have heard a lot about this. They're using it as cures for cancer, they're using it as cures for HIV, and they're using it as cures for birth defects in people. This technology overcomes many of the limitations of the traditional transgenic techniques that have been around for the past 20 years. These techniques have been difficult and cumbersome, and they involve the insertion of foreign DNA into the genome of the animal you're changing. This is why it's called transgenics. The first transgenic pig, Prrs-resistant pig, that Randy Prather made took 10 years to make. With this new technology, we can do it in 3 months. That's huge. So this is also known as genome editing. It's fast, there's no foreign DNA, and really, when you look at it, it imitates normal processes that might occur during evolution. If you were to stick a pig out into the wild and it was flooded with Prrs, eventually you would expect that it would become resistant. And this is a process of mutation. So in essence, we're just kind of short-circuiting about 2 or 3 million years of evolution in doing it in 3 months. This is how the system works. We have our gene of interest, in this case CD163. There are some guide sequence that tells these other scissor-like enzymes to cut the genome here. That piece of the genome is discarded, and then that same enzyme just sews it all up. Okay? That's how the technique works. In the case of how do we know we've actually knocked out this gene, well, we can look at it experimentally, and we have several tests that we can use. In this case, this is what our normal pig looks like in this test, and with this result, we know that we've actually knocked out CD163. So what I want to go to is a publication that goes back to last year, late last year, about a year ago. This is one of the highest impact journals in the world. This is the highest impact paper ever published on Prrs since its discovery. And this work was done at K-State. So I think if you want to look at the K-State team, you can begin to expand it, you know, and think of all of us as being a huge team. Just to kind of show you the results, if we take this CD163, we knock out this keyhole, what happens? Absolutely nothing. There's absolutely no virus present in the animal, and we don't detect antibody, and here is our Prrs affected lung, and then here is what happens if we take out CD163. So this is a 100% cure for Prrs. Pigs can't get it. When you ask the question, what do the pigs look like? Do they have 2 heads, 3 heads? You can't tell them apart. And as far as we know at this point, you know, their performance is fairly typical for a pig. In other words, this CD163, we can knock it out and it's not required for growth. And so what are our future directions? We think about this NBAF facility that's coming, and we're going to be studying all these exotic diseases. Well, the first thing we're going to try to do is to knock out those receptors to prevent infection. We continue to refine the CD163 knockout, And what I've done today is I've just scratched the surface. There's a lot going on right now that's going to have a profound impact on the industry. And then we have a list. We have a list of viruses, and guess what? We're going to start working down that list. And so it's just not going to be Prrs. It can be any other endemic or perhaps pig disease, emerging pig disease. So first of all, I want to recognize the National Pork Board who supported some of this work, USDA. Some of the genetic funding comes from PIC. And if there's one thing that I urge everybody is that all the greatest innovations in pork production have come through new technologies. This is a new technology, and they really need to be supported. I think right now we are really not being recognized by USDA and other funding agencies. So it's a good opportunity that if you ever meet a politician, tell them to fund pig research. And that's all I have. Thank you.